Good morning all. Good morning all to the FDP3, that is organized by Vishnu College of Pharmacy. So we are very happy to share that today the FDP is given by our renowned alumni senior Arjun sir. And then, so thank you all for joining in the meeting. And then over to Mohan Verma sir.
Dear staff and students, today we are having with us Arjun Rao. He is highly eminent alumni from SUCP. Earlier he did M.Pharm from SUCP and then he joined Aurobindo. At present he is in the Hetero Labs. He is highly experienced in the area of FR&D. So I thank Arjun Rao much for accepting the invitation. He will explain about the recent trends and about the innovations in the industry in this FDP. So I feel extremely happy to have Arjun Rao as a speaker. So I wish you all the best, and all the students and staff, you can take the advantage of this FDP. You can freely interact with Arjun Rao. Thank you, and overall thank you and wish you all the best. Over to Brahmaya sir.
Sir. So we are having Saloni madam. Madam will give introduction regarding Arjun sir. Thank you Arjun sir for joining in the meeting. Welcome to the meeting. Thank you so much. Over to Saloni madam.
So good morning everyone. I am Saloni, Assistant Professor from Shri Vishnu College of Pharmacy. Today I take privilege to introduce Mr. Arjun Rao sir. So Mr. Arjun Rao Panchali is a native of Vizag and completed his schooling in Jawahar Navodaya Vidyalaya, Vijayanagaram, followed by intermediate from Sri Chaitanya Junior College, New Gajuwaka, B Pharmacy from Aditya College of Pharmacy, Surampalam, and M Pharmacy from SVCP, Bhimavaram. Presently, he is doing his PhD in pharmaceutics specialization in Vignan University, Guntur as a part-time. After completion of M-Pharmacy, he had done his project work at Asian Drug Research Solutions Limited, Hyderabad. Immediately after completion of his academic project, he had joined in Aurobindo Pharma Limited Research Center in Formulation, Research and Development Division as a research associate and worked for almost 12.5 years for various regulated markets. There, he was involved in filing of more than 20 ANDAs for various regulated markets and filed 2 patent applications in USA and India. 1.5 years back, he was moved to Hetero Labs Limited to same FR&D department as a senior scientist too. He is handling a team of 5 scientists with almost 20 projects for various regulated markets. He had hands-on exposure working on all BCS class molecules and various categories of drugs, including NCE1 and para-IV filings. Thank you, everyone.
Thank you, Saloni madam, for introducing our senior. So now we are having our renowned alumni Arjun sir. He is a well speaker and he is a guide for so many juniors of Vishnu College of Pharmacy. So requesting Arjun sir to proceed over the section. Thank you all. Thank you for joining in the meeting. Over to you, sir, Arjun sir.
Okay. First of all, thank you for nice introduction. And I wanted to thank management of SVCP for giving this golden opportunity. And special thanks to Dr. Natraj sir and Dr. Mohan Verma sir. And thanks to Brahmaya for organizing this meeting. Am I audible to you boys? Brahmaya, am I audible?
Yes, sir. Yes, sir.
You're audible.
Right.
Yes, sir, you are audible.
Right. Thank you. So today I am going to present the trends in medical technology, innovation, and pharmaceuticals. So these are the contents I am going to present. The first and foremost is innovation with respect to input API and excipients, and innovation with respect to equipments and machinery. And the third one is the continuous manufacturing process, and followed by digitalization and robotics in pharmaceutical technology. And last one, the expectations from various regulatory agencies with respect to size and shape guideline and titanium dioxide free materials and nitrosamine free. So these are the contents I am going to present today. And in later sections, I am detailedly explain one by one. So the first one, innovation with respect to input API and excipient. For any drug formulation to prepare, we need active moiety along with some excipients. So previously they used to use a single API moiety in the formulations. Now the technology has been improved a lot, where the scientists are going to use API in the form of premix. So let us understand what is meant by premix. So to have a better bioavailability and to have a better absorption, the API is making as a premix along with some surfactant and solubilizer. So let it be quote with an example. So there is an evacaftor which is used to treat cystic fibrosis disease. So there the API evacaftor is mixed along with the ipramalose and SLS, a solubilizer and a binder, what we call it as, and as SLS as a surfactant. So together API, ipramalose, nothing but HPMC and SLS, they made in the form of premix, and that can be used in formulation. So if you take the bioavailability, if you compare the formulation which is made using a single API entity and what is the formulation made with the use of the premix, there is a lot variation with respect to bioavailability. The bioavailability of the formulation which is made by using the premix is more, and it is almost bioequivalent with respect to the NDA molecule compared to the formulation that was used as a single API moiety. So the thing is that whenever we are making an API into the premix, the bioavailability is increasing. Thereby, we are able to make a bioequivalent formulation with respect to NDA. So not only there are so many examples we can quote where the API used in the form of premix. So the second is the novel and new polymers. Even the excipient vendors are doing research on so many excipients, where thereby improving their results and producing excipients with a novel grades. So they are like spending more time on their R&D and giving the excipients which are superior quality with respect to existing excipients. Next is the co-processed excipient. So to prepare a formulation, we need to use a binder, surfactant, lubricant, and disintegrant, diluent, along with APIs and some other categories also. So now the technology has improved in such a way, the excipients itself, they are giving the it's a co-processed excipient means it is a mixture of three ones, like it is a mixture of diluent, disintegrant, and lubricant. There are so many formulations you can prepare by simply mixing API along with co-processed excipient. You will get a tablet. Imagine where the technology has been gone. So in the other way, we need to take the single diluent, we need to take the single disintegrant, you need to add the lubricant, followed by you need to go the compression. The shortcut thing is that you are getting a co-processed excipient where you can mix with the single API entity and will make a tablet. So the technology has grown such an extent that usage of co-processed excipient will enhance the productivity and give you a good yield. And the fourth one is the evaluation of the new grades within the same excipient. For suppose if you take the lactose monohydrate, so if you want to go for a wet granulation, there is a grade called as pharmacose 200M. So there it is in the form of a milled grade as micronized grade, where you can use only in the wet granulation process. So the excipient vendors have come up with a new technology where if you wanted to go for direct blending process or dry granulation process, the use of pharmacose 200M, which is a milled grade, is not at all acceptable due to flow issues. So actually they've done their R&D at their end, and they came up with some excipients with a superior flow where you can use the excipient in direct blending process. So the quality of the material has been not changed. They have done enhancement of the flow. Thereby, you can use the same excipient in direct blending process and roller compaction process. That means bringing up the new grades of excipients within the same category. So the last one in API and excipient section is low content of peroxides and nitrosamine grade excipients. So many guidelines has come to reduce the peroxide content and nitrosamine contents in the finished product. So we need to reduce in each and every step to which will contribute the peroxide and nitrosamine in the final formulation. So excipient vendors came up with new excipients with low peroxide content. Per suppose if normal limit for peroxide is not more than 20 ppm, there are excipient vendors who are giving same excipient with a limit not more than 5 ppm. So they are come up with a new technology and reducing their peroxide contents and nitrosamine in the input excipient itself. Thereby, the contribution to the final formulation will be reduced and we will get a stable formulation. So the next slide is innovation with respect to the equipment and machinery. So there are so many advancement and research has been done with respect to equipments also. Thereby, we can increase the yield and we can decrease the time of the process. So spray dryer, you may think the spray dryer is a long back process. I am just quoting about the spray dryer with lot of accessories, which is giving the more yield and completing the process with a lesser time. So for suppose if you are giving if you are taking 100 grams of material into a spray dryer, if the output material what you want, you are getting about 50 grams, there is no use because 50% of the material is going into the well. So nowadays, the equipment has came with some other accessories and other technologies which will ultimately increase the yield of the material. You will get a more than 80% yield. Thereby, you are improving the process by 30% compared to earlier process. If the materials like heat sensitive material, they are coming with the coolers and chillers. So thereby, you will avoid the heat resistant materials to undergo degradation, like thermolytic degradation. So in the similar way, like spray dryer, they have so much advancement takes place with respect to hot melt extruder. So both spray dryers and hot melt extruders are helpful to convert a crystalline form of drug into the amorphous form. Thereby, you can increase the bioavailability of the drug. So when you are going for the para 4 filing and NCE-1 molecule filing for a formulation, the innovator company may file it will patent the polymorphic form of a molecule. So let it be say crystalline form 1. So thereby you cannot use the crystalline form 1 drug in the final formulation. So in such instances, in such circumstances, you can go with the amorphous form of the drug. Thereby you can give the bioequivalent product of the NCE-1. So in the similar way, there is a lot of advancement with respect to bilayer and trilayer compression machines. Previously, they used to weigh the two different components, layer 1 and layer 2. The layer 1 contains a single drug and layer 2 contains one or the two drugs. They used to manually, the speed of the compression machine will be very less. Nowadays, the technology has gone in such a way that the productivity has been increased, that the time has come to half. The process takes 3 hours previously, you are able to get the same productivity within an hour time. Within a single hour, you can get that same productivity. So there is other machine called as tablet in tablet compression machine. It is amazing that you are getting a small mini tablet in other big tablet. So the machinery R&D has gone such an extent that it is able to give a tablet in tablet technology. And the other thing is the mini tablets in capsules. So we all know that many of the cases we fill powder or pellets in the capsule, whereas the technology has grown up with where we can accommodate a small mini tablets into the capsule. The number of mini tablets will be vary from formulation to formulation. So there may be 6 tablets or 8 tablets. So there are some extent where you can go with the 12 tablets in a capsule. So based upon the requirement, you need to fill the mini tablets into the capsule. Yes, some other instances you can fill both powder and tablets in a single machine, which will have a 2 hoppers In one hopper, you will fill the mini tablets. After that, it will go into the other hopper where you can fill the powder. Together, the capsule will be locked and it will come as a finished product. So the next technology is the liquid material filled in capsules. The example is the isotretinoin capsules, where you can melt you can use the nanonized form of the API isotretinoin, and you need to take a liquid melting a liquid or semi-solid solution with a semi-solid moiety, and you can fill that semi-solid or liquid form into the capsule. So this is a new technology that recently arrived, and so many companies are working on this. And the last thing with respect to innovation in machinery is packaging blister machine with disc and frozen. So to have a robust or stable molecule, so preparing a formulation is not at all acceptable. We need to produce the stability to ascertain the shelf life for a particular product. So you need to ascertain certain shelf life for a product for 2 years or 3 years. To ascertain that shelf life, you need to produce the stability data at different stability climatic conditions. So in HDB bottles, if you wanted to put in stability, you have a provision to put a discant like silica gel and everything. So in Bestro packaging, there is no provision to provide a discant like that. So nowadays, the technology came up with a Bestro packing with machines where there is a cavities for the discant. So the discant will be filled in that cavities, and that has been connected to the capsule blister pack. So thereby the discant will give an inert atmosphere to the product. Thereby the degradation will be decreases. Thereby the stability will be enhanced. So the next thing is the continuous manufacturing system. Nowadays, if you see many companies like Pfizer and GSK, to increase the productivity and to decrease the time, they have come up with the continuous manufacturing system. So what do you mean by continuous manufacturing system? So the first sentence itself, it is explaining manufacturing of a pharmaceutical dosage form without or a little manual intervention. So previously the manufacturing process at plant level or manufacturing plants will be like there will be a lot of intervention with respect to human being, that we need to sift the material, we need to load into the RMG, we need to do the granulation, we need to unload that material, we need to mill it, we need to again go for pre-lubrication and lubrication. After that, we need to go for compression. After that, film coating. So it is amazing to see a process like this without taking the material at any point of time outside. So recently, I have visited some other company and where I have seen the continuous manufacturing process. Since from dispensing to since from arriving of the film coating tablet, there is no manual intervention. So the like dispensing is done based upon the manual dispensing is done, and the materials will be given to the machine. It is going for sifting, it is granulating, it is drying, it is milling, it is blending. After that, compression and coating also done simultaneously. So you are giving the only raw materials as a required quantity, you are getting a finished product. In between, there are in-process control checks also. So in between, the machine is taking the material to see the LOD. So how much the moisture content that is. In between, it is taking the samples to check the blending uniformity and blend assay. It is like what are the in-process controls. In between, it is taking the samples to check the tablet weight and hardness. It is checking the thickness. So with predefined parameters, it is producing a finished product. So the same thing is possible for direct compression process, for wet granulation process, and dry granulation process. So I have seen the machines for direct compression and wet granulation process. The dry granulation process requires some manual interruption in between. So again, the machinery scientists are working to avoid that manual interruption with respect to dry granulation process also. So with respect to direct blending, direct compression and wet granulation process, the continuous manufacturing process reached a higher level. when compared with the dry granulation process. So the digitalization and robotics in pharmaceutical technology. So the technology is going the similar way. The pharmaceutical companies also gone in a similar way, where in previous days they used to note down all the experiments that are carried out in laboratories in manual L&Bs. They used to have a record, as like we used to have in SVCP, and like in all the colleges we used to have a record to note down our experiments. In the similar way, in industrial point, they used to have a L&Bs, manual L&Bs. Gradually, the manual L&Bs has been replaced with the eLNB, electronic laboratory notebook, where the management is providing the systems in like in manufacturing area only, where you need to conduct your experiments and you need to enter in that eLNB. So again, it is a like time, like you can reduce the time of experiments. Why? Because writing and note downing, just it will like it will save so much of time, and even there is a chances for losing of that manual LNB. You can keep it somewhere, and again you can forget that where I kept the LNB, and the archive is also very somewhat difficult. If you go with eLNB, although the things can be overcome. Even there is a provision to clone the experiments in eLNB. If you are going with a similar type of like experiment, you can clone with the previous experiment, so that it will give the observations, like not observations with respect to procedures, it will give in a predefined manner. So simply you can enter your experiments, like what are the observations you are going to the what are the changes with respect to previous experiment, what are the observations that are varied from previous experiment, that you can simply enter in the ERNB, so that you can save so much of time, and you can use the same time for other compilations and everything. So the second one is the manual hard copy entry to system entry. So even at plant level, they use it to write all the hardness and thickness and in-process controls in a manual hard copy. This is called as BMR, batch manufacturing records. So they use it to check in-process control tests like tablet weight variation, friability, hardness, thickness, and everything, disintegration and everything. They use it to write down manually. Nowadays, they are going with the system entries. So the next is the predefined program setting in machines. So there is a digital screen for every machine, let it be a compression machine or coating machine. If you are giving a value for that, the machine is automatically setting that weight. So like pre-compression forces, the tablet weights, tablet thickness, and everything predefined, and need to give in a systemic like in system you need to like load that recipe. So the machine will run accordingly as per that recipe what you are going to loaded in that system. So nowadays the equipment has been came with that recipe systems. The fourth one is the use of robots in material arrangement and pickups. So I have seen in two or the three companies where robots are used to arrange the materials in warehouses. Where as per the R&D is concerned, we will use the like grams and kgs of material only. It is like 250 grams, 500 grams, or 1 kg like that. If you go to the plant level, there will be tons and tons of material need to be take the commercial batches. So again, lifting the same material with cranes and all will take some more time. So I have seen a robots for placing that material in the warehouses. They just as a program, they are giving the programming to actually to the robots, where they are telling that we need to keep the material of so and so batch number in rack number A6. It is going straight to the A6, and it is simply placing that container in that respective place. Again, whenever you wanted to dispense the same material, if you give the program to that robot, it will go there and bring that material and give it that balance. You need to weigh that material, again you need to send it through robot. So how much it is how much time it is saving? So it's a lot time is saving when we are using the robotic technology in the manufacturing system. So I don't know apart from warehouse where we can use the robot. I have seen with my naked eyes where the use of robots in warehouse of 2 to 3 companies. So as I have already mentioned, the use of technology in performing the in-process quality tests. So in the continuous manufacturing system, as I stated earlier, while doing the compression, we don't know what weight it is going to like set and with what weight the tablets are going to be evolved out. So to check that, we need to have we need to perform the in-process quality test like how much tablet weight it is, how much hardness it is having, how much thickness it is having. And before that, in wet granulation process, after drying, how much LOD it is having. So if it is having the less LOD, you may get sticking and picking tendencies during compression. If it is like over dried and if it is having the less moisture content, there is a chances of capping and lamination during compression. So to avoid all those things, we need to fix a LOD limit during drying. So you need to fix the LOD limit in the recipe. So by checking the in-process LOD at that time, it will give the clearance to next step. So for suppose if for a product, if you fix the LOD in between 1.5 to 3%, it will the drying will be takes place till our LOD is reached in between 1.5 to 3 only. If it is 1 point less than 1.5, more than 1.5, it will not allow. And if it is more than 3 also, it will not allow. So thereby you will have a smooth manufacturing process till the film coating. So the last slide, what I am going to explain is expectations from regulatory agencies. So you are making the product at R&D level, you are taking that into the plant level. You are taking the exhibit batches where you are submitting the samples for pivotal bio studies. Your product is bioequivalent, and you are keeping the exhibit batch samples in stability, and your product is stable. You can ascertain a shelf life of 2 to 3 years for that product. Ultimately, we need to produce all this data to regulatory body. So whatever the regulatory body it may be, if you wanted to file for US, you need to produce the data for US FDA. If you want to file for Europe, it is MHRA or EMEA. If it is for Australia, you need to file it for TGA. If it is for Canada, you need to help for Health Canada, ANVISA for Brazil, PMDA for Japan. So there are so many regulatory agencies where whatever the data we are producing since from R&D development to exhibit batches, we need to produce to regulatory agencies. Nowadays, the requirement of agencies has been grown like anything. So since they are interacting with so many companies and so many MNCs, so they are expecting the same thing from even for the similar and smaller companies also. Since they have more interaction with the more MNCs like Pfizer and GSK, the quality will be more. In the similar way, what they are producing, their expectations from smaller companies also in a similar way. So there is a guideline called as Size and Shape, which has almost in 2019 or 20 it has been published. So I can simply explain what it is size and shape requirement for US market. So for ease of swallowing by a patient, the agency came up with a guideline called as size and shape guideline for tablet dosage forms and capsule dosage forms. So there will be 3 criteria with respect to size and shape guideline. So for the tablets which are having largest diameter 8 mm, so you can go with the like for suppose I am telling about the reference product or NDA. So we need to compare the size and shape of our generic or AND products with the reference products. So if the NDA is having 8 mm or diameter or less, you can go in line with the same similar diameter for your AND or generic product. If the NDA is having the size larger dimension, it is having the 8 to 17 mm. You can deviate with respect to NDA with respect to 20% with respect to size and 40% with respect to volume. If it is the largest dimension of the tablet is in between 8 mm to 17 mm. If the largest dimension of the tablet is more than 17 mm, the agency is not accepting ANDA to go beyond that. I mean to say largest dimension is if it is the capsule size, it is the diameter is the largest dimension. If it is a oval shape, if it is a round shape, it is almost a diameter only. The largest dimension, why I am mentioning about, the largest dimension is nothing but diameter of a capsule shaped tablet, otherwise oval shaped tablet. So if we are the NDA product or reference product is having more than 17 mm, let it be 19.5 mm. So your generic product should not cross 19.5 mm. It should be 19.3 and 19.2. 19 is allowed. Beyond 19.5 mm, agency will not give you an approval. So up to 17 mm, there is a provision that you can deviate up to 20%. For suppose 10% means 10 into 20% means 12 mm. If the RLD is 10 mm, you go up to 12 mm. But if it is crossing 17 mm means not at all you are allowed to deviate with respect to NDA product. Coming to the capsule, the highest size of the capsule that can be used for human use is 00 size. So there are capsule sizes from 4, 3, 2, 1, 0 and 00. So beyond 00, the agency is considering for a veterinary use. So there is a capsule size with 000 also, which is used to for human, not for human consumption, it is used for veterinary purpose. So some deviations are allowed. I will quote an example of cefixime capsules, which is an antibiotic capsule, which is produced by Lupin Pharma. So there the reference product is having 00 elongated EL size. So with a proper justification, you can go with 00 EL also. If the other generics are having the same dosage form, which is bioequivalent with that of the Lupin cefixime capsule with a size 0, the marketability will be more. So people or patients will always think of the patient compatibility and ease of swallowing. So people will feel somewhat difficulty in swallowing the capsule of 00 EL compared to the capsule of 0 size. So A&E like generic companies are coming with the novel ideas to decrease the size compared to the reference product. So I have done a like same cefixime capsule I have developed in one company in my previous organization with size 0. I made it bioequivalent and filed for US market. So the second thing is that titanium dioxide pre-formulations for film coating materials. So nowadays, the Europe regulatory agents has thought that titanium dioxide is a carcinogenic and mutagenic. So it is simply it has given a statement and it has produced a MHRA statement by saying that what are the dosage forms that you are giving for Europe market, that should be titanium dioxide free. So titanium dioxide can be used as a opacifier, and many of the coatings, if it is a white coating without opacifying nature, you can produce a film coating. So Europe is strictly telling that we don't need the formulations where we are using titanium dioxide. Nowadays, the excipient companies and pharmaceutical companies came up with an alternative for titanium dioxide. They are using magnesium oxide and calcium carbonate as the alternative to titanium dioxide. Actually, it requires somewhat, if we are using the titanium dioxide as a opacifier, 3% film coating is sufficient to give you a uniform coating. If you are going with the other calcium carbonate MgO base, it requires 5% film coating. So titanium dioxide is good opacifier. So even the scientists and regulatory companies are explaining the agency that titanium dioxide is not that much carcinogenic, and they are telling that we will produce a limit, beyond that we can't go. So don't know, the discussions are going on at regulatory agency levels. We don't know how it will be end up. So even US market, they are not raising any concerns. The titanium dioxide is only raised by MHRA and EMA, only European regulatory agency. And the next thing is the nitrosamine free and low nitrosamine impurity formulations. Nowadays, the most trending and most thing is that nitrosamine in all the companies. So it has come, it actually, it came into the picture when they have used the sartans. So all valsartan, losartan, olmesartan, telmisartan, all antihypertensives. So they came to know that it is having nitrosamines. Again, these are the carcinogenic and mutagenic. As these are used for the antihypertensive, the usage will somewhat longer period. So upon longer period administration, they're telling that it may produce a mutagenic activity and carcinogenic activity. So that's why the USFD has banned so many things, so many formulations of certain products. So again, the excipient vendors came up with the excipients which are having the low nitrosamine content, and there are otherwise there are so many excipient vendors which are giving the nil nitrosamine. They're like what are the excipients that they are giving? It's not at all containing any nitrosamines. So much research work is going with respect to nitrosamine evaluation. Method developments like ICP-MS is required to detect that. So many methods we need to check the interference of the other excipients for this nitrosamines. The example for nitrosamines is NDEA, NDMA, NDIPA. There are 7 main nitrosamine impurities are there. Actually, previously, when at the initial time point, they used to give us low level 96.6 nanograms is allowed for NDMA. Again, lot of research work has been done, and for some nitrosamine impurities, the level has been reduced, gone up to increase it to 1500 nanometers also, like 1500 nanograms. Sorry, not nanometers. It is a nanograms. So it from 96.6 nanograms to the level has been increased to 1500 nanograms based upon extensive research. So now in every company, more research work is going towards the nitrosamine or low nitrosamine content formulations only. And the last thing is control of mutagenic impurities as per ICH M7 guideline. So during preparation of the API or active moiety itself, there is a chances of carryover of the mutagenic impurities. There is ICH M7 guideline which explains clearly how much limit we need to fix for that mutagenic impurities. So you can also change the intermediate and the starting raw materials of the API to avoid these mutagenic impurities. If these mutagenic impurities are unavoidable, so you need to fix a limit by using that ICH M7 guideline. So in a single nutshell, I can say the industry are like the work at industries are going like anything, and the thinking attitude of the scientists who are working in industries also like increase, and they are simply working on a working in an efficient manner and to produce a stable and robust molecules. So with this, I wanted to end my presentation. If you have any concerns, if you have any doubts, just I wanted please let me know. I will explain.
So, Arjun sir, thank you so much. Dear all, any doubts are there, please can you message or can you ask the Arjun sir.
Guys, I'm expecting at least 2 to 3 questions here.
Any questions from participant, you can ask the Arjun sir. So someone is asked on YouTube, what is para 4 file?
So there will be filing called a para 1, para 2, para 3, para 4 filing. There is NCE minus 1 filings also. If we are there will be a patent filed by the innovator company or some other companies by restricting the use of polymorphic forms or excipients. So we need to non-infringe that patent. So whenever that patent has been filed by a company or something agency, if it is in application form, we need not to bother about. If it is in patent, if the patent is granted from patent office, we need to avoid all those excipients what was claimed in that patent. Again, there will be like dependent and independent patent claims. The independent claim has a more attention compared to the dependent claim. The claim number 1, 2, 3, which are independent, have a more attention, and we need to avoid that. So you need to go like patent. The patent may ask to avoid the usage of the process. It may use it to. It may actually claim to avoid the particular polymer. So in some cases, it can claim to use the percentage of the excipient. For suppose you need not to you should not use the HPMC as a binder from 5 to 15%. So he has done a extensive work from 5 to 15%, and beyond that concentration, from his perspective, we cannot produce a bioequivalent formula. So in such a way, he will fix a limit and he will claim the patent. So in para 4 filings, where we need to non-infringing the patent, we need to avoid all those patent claims, and we need to produce a bioequivalent formula.
Thank you, sir. Thank you, Arjun, sir. Any doubts? To someone, sir, thank you for providing knowledge and industrial exposure. Thank you, sir. Any questions? Any questions? Here all, any questions are there? Can you please ask Arjun, sir? I think you explained well, sir. Maybe there is no questions from their side.
Okay, if it is the case, like I wanted to once again thank management of SPCP for giving this opportunity, and special thanks to Dr. Nataraj sir and Dr. Mohan Verma sir and the organizer Brahmaya. Thank you. Signing off, right? Thank you.
Thank you, sir. Thank you, Arjun, sir. You have given a brief explanation regarding innovations related to the API and excipients, and guidelines that are related to the peroxides and nitrosamines, etc., etc. And then what are the innovations that are going in respect to the machinery and equipments in industry, and also what are the different technology development that are taking place in the R&D and processing, etc., etc. And then digitalization and robotics. So now one of the most important parameter, that is digitalization and robotics, the importance of digitalization, importance of robotics in pharma industry. And then what are the expectations that from the regulatory agencies. So you have given a well explained manner, and thank you for sharing your knowledge. And we are very happy to share that Sir is our alumni, and Sir is guiding to so many juniors like us and like so many our friends, etc., etc. Thank you all for joining in the meeting. Thank you so much. Thank you once again. Thank you. Dear Arjun, sir, thank you once again for joining in the meeting. Thank you, sir.
Thank you, Brahmaya. Thank you. Signing off, right? Thank you.
Thank you, sir. I'm ending the meeting. Thank you all.